Hello, my name is Kayla Marie Jones, and I will be presenting to you about biogas production from co-digestion of corn stover and chicken manure under anaerobic wet, hemi-solid, and solid-state conditions. I will be representing the Department of Agriculture and Environmental Sciences at the Tuskegee University. The outline will be as follows: the introduction, objectives, materials and methods, the concept map, results, and references. So we would like to open up the introduction by defining anaerobic digestion, which is a process in which bacteria breaks down organic materials in the absence of three materials, that is concerning oxygen, carbon dioxide, and hydrogen sulfide. All of those materials that are taken from the substrate and inoculant will basically produce the methane that we'll use to power homes, cars, and other electronics. And we would like to also say that there will be three conditions, which will be the wet condition, the hemi-solid condition, as well as the solid state condition. There will be three types of different reasons or different factors that we're trying to yield the methane, but we'll talk about these three. So in definition, the methane is an odorless, colorless, flammable gas, and it is or can be found in a stove or a gas stove in a apartment. And lignin is a substance found in wood, and it is found in the cells walls of plants. And then the last one is cellulose, which is a renewable carbon source, and that can be found as vegetable fibers or cotton. So this is the introduction where the corn stover is the substrate, and it has leaves, stalks, and cobs of the maize, while chicken manure is also the substrate, and it has chickens, which are basically using the feces of it as an organic fertilizer, and that's usually low in nitrogen. So both produce methane, and they break down the lignolytic as well as the cellulolytic components of the substrates. The objectives are that the corn stover to the chicken manure ratio will process under the wet state, the hemi-solid state, and the solid state condition. We're hoping that this will yield high methane production, and we'll use the BMP assays, which is the biochemical methane potentials. We'll investigate it further with the volumetric methane productivity, and we'll be using this with the factors mentioned, which is the three conditions and the inoculants. The first step is hydrolysis, which is the breakdown of the chemicals. So it could be such as the oxygen, or it could be hydroxin. And the second step is acidogenesis, which uses the process of fermentation, and that also helps convert simple molecules into a methane byproduct. The third process is acetogenesis, which is a little different. It produces the acetic acid, and that's also concerning the carbon dioxide and hydrogen. The last step is methogenesis, and that's basically to show you the metabolites and the previous steps used by bacteria to make carbon dioxide, methane, and water, as we talked earlier. The final products of all of this process is methane, carbon dioxide, and hydrogen sulfide. So hopefully this will reiterate how we're going to power or heat homes and use it as a biofuel for fueling vehicles, as well as producing electronics or electricity for electronics. And here is basically a stage showing hydrolysis, acidogenesis, and acetogenesis, as well as methogenesis. The flow chart basically shows you from the complex polymers, the monomers and oligomers, to the propionate and bitrates, and then it also tells you what was broken down and that's to basically make the carbohydrate as well as the methane. So this is basically the methods and materials part of what was used in the study. The Hock assays as well as the kits were containing the total alkalinity rate, and they also had us use titration and sulfuric acid. They had basically yielded the perimeters, which were the pH balance, the total solids, and the volatile solids. And below is a picture of a gas analyzer, and it was used to basically determine the composition, which are the chemicals or elements in the biogas, and that was from the reactors. The concept map here shows you basically what we do for or concerning the materials and methods. So one is to obtain the chicken manure and corn stover. Two is to conduct and observe. Three is to characterize. Four is to test for the methane, lignolytic, and cellulolytic properties. And five is to evaluate the methane that was yielded from the reactors. And here are two pictures. The top picture is the chicken manure, which was retrieved from Beijing, China, and that was kept at 20 degrees Celsius. And then we have on the bottom the corn stover, which was found on a chicken farm at Beijing, China, and it was basically both of these were used in the inoculant, and this is how it looks before it is broken down. So the results are as follow. The corn stover was contained higher in the total solids, which is the TS you see there, and the volatile solids, which is the VS you see there. And that is of the chicken manure. The volatile solid to total solid ratio was 94.1% and the 78.1% for the chicken, or excuse me, corn stover and chicken manure. So that means the carbon to nitrate ratio of the corn stover and the chicken manure was either too high or too low for the anaerobic bacteria to basically grow and to yield methane. And this is some more results basically showing the significance of the methane yielded in the volumetric methane productivity, and it's concerning the three conditions, which shows that co-digestion can increase the methane yield, and the highest methane yield was 218.8 milliliters per grams, and that's of the volatile solids added, and the corn stover to the chicken manure ratio, which was 3 to 1, and that was in the best wet state of the anaerobic digestion. So that follows with the last point being the highest volumetric productivity of 14.2, and the L-methane to the L-reactor volume occurred in the SSAD, which is the semi-solid anaerobic digestion condition, and that was to the corn stover and the chicken manure ratio 1 to 1. And the volatile fatty acids to the total alkalinity value, which was 0.4, and was more important due to evaluating the process of stability of reactors for the future. And here are the references. And if you have questions, you can send me either through email, which is kjones6918@tuskegee.edu, or you can also provide questions below here in the comment section. Thank you, and I hope that I provided you with some knowledge. Have a good day.